Good to see you all. I'm Goo Lee from the Department of Pharmacology. Today, we're going to talk about treatment of so-called protein conformational diseases by activating IRE1 alpha, and more specifically about treatment of cystic fibrosis and Pendric mutation-caused hereditary hearing loss, and development of therapeutic medicines to treat those diseases. First of all, in my lab, we're studying how substances are transported through membranes. I don't think the remote controller works, but anyways, in these cellular membranes, we've got transporter proteins that transport substances. In my lab, we're mainly researching those cells in epithelial cellular membranes in digestive and respiratory systems where, as illustrated in the figure over here, transporter proteins like SLC solute linked carrier, ABC, and ATP binding cassette transporter exist. Well, as for these proteins, as you can see in the right-hand side of the figure, it is known that these proteins are known to be generated in endoplasmic reticula, or ER, and once generated, proteins head to cellular membranes via Golgi. This is the very famous and important concept in the Society of Cell Biology, and scholars having researched this concept have won several Nobel Prizes in physiology or medicine. In the Society, we use the term conventional trafficking to refer to transorganic transfer of these proteins off the ER to Golgi and to cellular membranes, as illustrated now on the very left. While a number of cells work to transfer these proteins to cellular membranes via conventional trafficking in peacetime, proteins reach cellular membranes by passing Golgi when cells are stressed out too much. As you can see on the right, these proteins in red bypass Golgi, reaching cellular membranes, which is what we call unconventional trafficking. What we discovered is that we can therapeutically capitalize on this unconventional trafficking, so let me tell you how. Take a look at the example where the conventional trafficking is illustrated in terms of how proteins move transorganically. Once these proteins are generated in ER, the way I've explained above, they are quality controlled also in ER to make sure they are of acceptable quality. If you have understood what I have just said, it would be quite reasonable to call this as a quality control process, which is the way this process is actually called. We call this process as ERQC, standing for ER Quality Control. Once properly generated as determined in ERQC, these proteins head to cellular membranes to transport substances. However, the defective proteins, defective in terms of protein conformation, are sent back to the recycling plant to proceed with the ERAD, standing for ER-associated degradation. Almost all such defects are congenital and causative factors a variety of genetic diseases, as these proteins, which are supposed to reach cellular membranes to serve as intended, are degraded in the middle. One of such genetic diseases is cystic fibrosis, the most lethal genetic disease around the world, and is more common in Western populations than Asian populations, like Koreans. And another such genetic disease that is more commonly contracted than other lethal genetic diseases is Pendric Syndrome, which is more commonly manifested in Korean populations, not in the form of a syndrome, but in the monosymptomatic form of hereditary hearing loss, attributable to a specific cellular membrane protein called pendrin failing to reach cellular membranes. Hereditary hearing loss occurs when this failure happens, most commonly in pediatric patients. In Korea, the most common cause of pediatric hereditary hearing loss is mutation of pendrin. Here's why things are getting interesting. These mutated pendrins that causes hereditary hearing loss, or the mutated proteins that cause cystic fibrosis, called CFTR, somehow work if they reach cellular membranes as they were supposed to, if not mutated. Even more interestingly, these mutated proteins help treat those diseases when reaching cellular membranes. So this is why researchers around the world try to work these mutated proteins their way into cellular membranes to make them help treat diseases, as illustrated in the right. We've been working with this for 9 years. Back then, we discovered for the first time that the mutated proteins that fail to reach cellular membranes find their new ways into cellular membranes when the conventional pathway, trafficking off ER to Golgi, as mentioned before, is exogenously made unavailable. And we further discovered the genes called GRASP serves an important role in helping mutated proteins finding their new ways. For instance, in the non-clinical research using mice illustrated in the left, we've induced conformational defect in CFTR on the very right mouse to manifest the symptom similar to human cystic fibrosis. This mouse is supposed to die way earlier. In the third mouse from the left, we induced this overexpression of GRASP in CFTR and found out that this mouse grew very well, even to the extent matching the normal mouse, contrary to the mice with the conformational defect in CFTR where they died within 2 months. The implication of this research is that we now know the newly founded unconventional pathway to cellular membranes help the diseases to be cured. Even though such a simple method applied to those genetically modified mice cannot be applied the same way in human beings, what we found in the follow-up research is to use such method in human beings is that we recently found a protein called IRE1 serves a very important role. IRE1 sometimes serves as kinase, while serving as endonuclease in some other times. So, in our attempt to identify which type of IRE1 service is more important in curing the diseases than another, we found out in the first hand that IRE1 was not effective in paving a new unconventional pathway into cellular membranes when serving as an endonuclease. So we went on to see if that same IRE1 is effective when serving as kinase, and voila! We found out that the IRE1 was effective in establishing a new unconventional trafficking into cellular membranes. This is a simplified figure that illustrates our attempts to induce mutations in relation to kinase. So now that we knew IRE is effective in sending mutated proteins to cellular membranes when serving as kinase, we went on to see if exogenous activation of kinase helps curing the diseases. On the left, we see the clear evidence that the substance called CSTMP activates IRE1 kinase. On the top right, we see that when CSTMP is applied, the mutated proteins that were failed to bypass in the first place gradually take on their way to cellular membranes as time passes. We then began to think that CSTMP might help send the mutated proteins to cell membrane, and devised another non-clinical study you can see on the left to see if the idea is viable in the mouse models. I'm afraid I cannot point what I explain, but let's see. On the very left, you can see that CFTR exists in the apical membranes at the crypt cells of the colon, as the arrow points. And you can also see in the mouse model that expresses the disease CFTR does not exist in cellular membranes. In the mouse models that are indicated as F508del at the bottom, you can also see CFTR was expressed in the cellular membranes as stained green by the effort of IRE1 kinase activator, namely CSTMP. In terms of chloride channel, CFTR engages in transport of chloride ions. In light of the fact that impaired chloride channeling keeps respiratory and digestive secretions from being secreted in human beings and causes diseases. On the top left in the right pane, you can see the normal CFTR activity with normal chloride channel in a healthy mouse. And on the bottom left, you can see chloride channeling is impaired in the disease model mouse. On the bottom right, you can see the disease model mouse successfully recovers chloride channeling by the effect of CSTMP, So we now have an evidence that CSTMP may potentially cure the diseases. As I told you in the first place, we can assume that CSTMP can basically be used to cure a variety of diseases that cause folding defect in membrane proteins. With priorities on, as we expect, cystic fibrosis and Pendrin mutation caused hereditary hearing loss. The tables you see in the figure describe marketability of a variety of orphan drugs, and the cystic fibrosis drug is ranked number 8, despite its unreliable efficacy, frankly, entailing more than $3 billion or 3 trillion Korean won. Terms of Korean won, backing its decent marketability. In the next few years, we expect that the cystic fibrosis drug will be ranked somewhere around number 2, excluding those anti-cancer agents for rare cancers such as pancreatic cancer. And we also expect by that time, the market size for cystic fibrosis drugs will be about $7 billion or 8 trillion Korean won. This is why we now aim for developing a low molecular weight compound that effectively activate a new unconventional secretion or unconventional trafficking to cure those protein conformational I mentioned today. The prototypical CSTMP currently available is quite toxic, and as far as potency, EC50 sits around just 1 micromole. So we view that if we develop a new compound that is more potent and less toxic than the prototype, that would be a highly viable option to cure those diseases. The related paper was published on the February issue on Science Advances, and the domestic patent was applied last year. Also applied was a patent related to PCT just a few days ago. This is it. Thank you.